Good day everyone, and today we're going to talk about DNA replication process. DNA replication process is the biological process of producing two identical replicas of DNA from one original DNA molecule. In order to have a successful DNA replication, there must be a requirement in DNA replication. So first of it is the substrate used in replication. The DNTPs or Deoxyribonucleotidetriphosphate and the RNTPs Ribonucleotidetriphosphate is a form of energy that provide, initiate, and sustain the synthesis at the replication fork. DNTPs is the one that synthesizes the DNA molecule, and while the RNTPs synthesizes the primary molecule. The second requirement is template. So, the DNA must possess the fidelity of base pairing. Thymine is only paired with adenine, and guanine is only paired with cytosine. And for the third and the last requirement, which is the enzymes and other proteins. So, there are 7 necessary enzymes that are used in DNA replication. First is the initiator protein, marks a region of replication and creates initial opening in the DNA. The second is the helicase, the one that unwinds and separates a DNA strand. The third is the RNA polymerase, the one that creates primary molecule or RNA segment. And the fourth is a single-stranded binding protein, stabilizes and protects the newly opened single from reannealing and degradation from other enzymes. And the fifth is the DNA polymerase, the one that attaches a free DNA nucleotide to the growing strand, proofreading repair, removes and replaces primary molecule with the DNA nucleotides. The sixth is the topoisomerase, the one that removes torsional tension by cutting and unwinding the DNA strand, and the last one is the ligase, and the one that creates phosphodiester bonds between DNA molecule, replacing the primary molecules and its adjacent segments. So that is the essential and the necessary enzymes and proteins used in the replication. And now for the step in DNA replication. So there are 4 different DNA replication steps. First is the initiation. So the initiation start at the origin of the replication where initiator protein binds at the origin of replication, creating tension in the adjacent nucleotide. The tension disrupts the hydrogen bond, the DNA molecule slightly open up, then proceed to the second step, which is the unwinding. Unwinding the initial break or nick provides access to helicase to further unwinds and separates the DNA double helix by breaking the hydrogen bonds between each base pair. Unwinding moves towards together with the replication fork. Helicase moves towards the replication fork. They expose the single-stranded DNA are the unstable because they are prone to reannealing degradation caused by the nucleases. As you can see, the gray color in the image is the one that's what we so-called the enzyme called a single-stranded binding protein, the one that prevents and protects a single-stranded DNA from reannealing and degradation caused by the nucleases enzyme. As the helicase continuously unwinding or breaking the hydrogen bonds, the topoisomerase takes its place. It is the one that controls and reduces the supercoiling and torsional strain that builds up ahead of replication fork. So there are 2 types of topoisomerase, which is the topoisomerase 1 and topoisomerase 2, which differs in the number of strands that they will cut. Topoisomerase 1 cuts only single strand, while the topoisomerase 2 cuts the 2 strand in the DNA, and it will remove or uncoil the knots and then reconnect it to the DNA. The third step in DNA replication is the elongation, which involves most of the enzymes during the DNA replication process. So there are 3 steps in these steps in replication. So the first step is the formation of the primary molecule or the primase. It will the one provide 3' OH group of the for the attachment of the DNA nucleotide to start the elongation process. So as you can see, the green color there in the image is the primase or the primary molecule. For so the second step of elongation is the very important step where the attachment of free nucleotides or pre-DNTPs to the growing strand. These performed by the DNA polymerase. So as you can see, the gray color or the white color with a big letter G is the DNA polymerase. So this is the one that attaches the 5' phosphate group of the first succeeding DNTPs into the 3' OH of the primer or the last nucleotide in the growing strand respectively. So the DNA polymerase synthesizes DNA in the 5' to 3' direction of the growing nucleotide strand. This process simultaneously takes place in the both strands. Okay, so the elongation process initially produces a 2 strands. So the first is the leading and the second is the lagging strand. So for the leading strand, the direction of it is from the 3' to 5'. So these strands spend the least energy as it only needs 1 primer to complete the replication process. While the lagging strand, the direction of it is from the 5' to 3' direction, and it needs more primer as it gives against the movement of the replication. And for the third substep in the elongation step of replication is the primer removal. So the primer in the both lagging and leading strand are removed and replaced with DNA nucleotide by a specialized DNA polymerase, or to be exact is the DNA polymerase 1. So the removal of the primer along the lagging strand exposes the stretch of the unconnected DNA fragments known as the Okazaki fragments. So as the primer molecules replace with free DNTPs nucleotide, there is still a nick or a gap between the first and the last DNA nucleotide added. So the nick or the gap will be sealed by the enzyme called the ligase. So as you can see the blue color in the image, in the last part of the leading strand, lagging strand, is the DNA ligase. So the DNA ligase is the one that creates a phosphodiester bonds between the 5' phosphate group of the first nucleotide to the 3' OH group of the last nucleotide of the two adjacent Okazaki fragments or newly formed DNA strand. So this DNA ligase seals the nick and the gap with phosphodiester bond. So now let's proceed to the last step of DNA replication, which is the termination. So there are two modes of termination process. First is when the two replication fork meets, and the last is the presence of termination process. Okay, as a summary of the DNA replication, so there are 4 steps. So the first step is the initiation, the second is the unwinding, the third is what we so called the elongation, and the fourth is the termination. So as this process moves around or goes around continuously, so there are two products for the eukaryotic DNA replication, or what we so called the two linear molecules of DNA. So that would be all, and thank you.